Good morning all. Today's topic for discussion is endodontic microbiology. As we all know that the dental pulp is a living tissue which is comprising of neural and vascular tissue, and any infection, we all know that in the body any infection is usually caused by the microorganisms. So similar to that, so even the dental pulp is also amenable to various infections, and the microorganisms pose a challenge to the dental infection. So the endodontic infection, let us go into the topic. So endodontic infection is primarily caused by microorganisms. So before we want to understand what are the various types of organisms which are present, so the first foremost is aerobic organisms. That means the organisms which can survive and grow in an oxygenated environment are called as aerobic organisms. And then next comes is anaerobes. Generally anaerobes are the organisms which can also survive in the absence of oxygen, depending on whether they can survive on complete absence of oxygen, or they can plus or minus without the presence of oxygen or in the presence of oxygen. They are classified into two various types, that is facultative anaerobes and obligatory anaerobes. Facultative anaerobes may use oxygen but also have anaerobic methods to have energy production. That means facultative anaerobes are those which can grow in the presence or absence of oxygen. Obligatory anaerobes are those organisms which grow strictly in the absence of oxygen. So these are the different types of organisms depending on the oxygen presence. Now when we see the portals of entry, so how do the microorganisms have an entry into the tooth structure? So the various modes of or portals of entry where the bacteria enters into the tooth. So the first one being dental caries, you have the periodontal tissues, you have the trauma, and you have the anacarysis, right? So dental caries, we all know that the dental pulp is encased in the closed cavity surrounded by dentine and enamel, right? So the bacteria, when it enters, the bacteria will have the access from the oral cavity via the enamel and the dentinal dentin. So dentin will have dentinal tubules which are viable from the dentin surface to the pulpal tissue. So any bacteria which enters into the dental tubules will have direct access, right? So that is one way of entry of the bacteria. Next is the periodontal tissue. Periodontal tissue, we can see this is a gingival crevice, right? So the bacteria, so when it is there in the gingival crevice, can enter into the tooth structure via the means of radial canals or the apical canals or the apical foramen also. Trauma, sometimes because of the trauma there will be complete exposure of the enamel, dentin and pulp that causes the exposure of the microorganisms directly to the pulp. Next is anacarysis. Anacarysis is nothing but it is the spread of infection via the blood. So what happens is usually in healthy individuals also sometimes we can see bacteremia, that is presence of bacteria in the blood. So for example, let us say that this is gingiva. So in the gingiva you can see that there will be some the gingival blood vessels, right? So which may harbor some sort of bacteria in them, and when these the microvasculature present in the gingiva, when it gets ruptured, then that gets access to the bacteria that can also go into the apical foramen and cause infection. So that is anacarysis. Anacarysis is spread of infection via the blood. Moving to the next topic, which is pathways of infection. As I already told that enamel and dentine are protective layers of the pulp. So once the dentine is exposed, so you have something called as dentinal tubules. Dentinal tubules vary in the diameter of 1 to 4 micrometers, right? And we all know that the bacteria size is hardly 1 micrometer. So now you can see here, these are the dentinal tubules. These are the dentinal tubules. So the bacteria can enter into the dentinal tubules because the size is very less compared to the size of the diameter of the dentinal tubules. They can get easy access into the dentinal tubules and reach to the pulp. Especially if the remaining dentine is less than 0.2 mm, then the bacterial access is more easier than compared to more remaining dentine thickness. So this is the pathways of infection. Next comes the open cavity. So the open cavity is nothing but when the tooth undergoes some trauma where there is direct exposure of the pulp because of the traumatic origin, or for example like a coronal fracture. So you have that is open cavity, right? Otherwise during the treatment procedures, if the operator is not following the aseptic technique, that means the operator itself, he himself is incorporating some sort of bacteria. So that leads to the access of the bacteria into the pulp. So these are the causes or the routes of portal of entry of the bacteria into the tooth structure. Right, periodontal membrane is already told. The tooth will have lateral canals and apical canals. So from the periodontium, the bacteria can traverse and go into the apical area and reach the pulp tissue. Right, and during the prophylaxis also, sometimes what happens is when you do during the prophylactic procedures, we force the bacteria into the lateral canals or the apical canals, especially in cases of periodontal pockets. This is one mode of entry. Next is bloodstream, that is called the anaclesis. I said the bacteria from the gingival crevice or the periodontal pocket may reach the root canal of the teeth, whose pulps become necrotic after trauma through severe blood vessels of the periodontium. Right, so these are the various portals of entry of the microorganisms into the pulp. And one of the reason is again faulty restoration. Here in the image we can see that this is the endodontically treated teeth, and you can see the post-endo restoration here, which is not properly filled, right? And the margins we can see this. So when the margins are not properly sealed, when the coronal restoration is not properly sealed, what happens? This will mark passage of entry for the bacteria. So here we can see that because the coronal restoration is faulty, the bacteria or the bacterial byproducts can seep into this root canal area and also cause reinfection in the cases which are root filled already. So it is said that usually when there are faulty restorations, the bacteria may take around 5 to 6 weeks of time to enter into the root canal treated teeth. Right, so this is also of significant importance. Now coming to the microorganism relation to oral cavity, there are almost around 700 species of microorganisms in the oral cavity, and endodontic infection as such is not a single infection. That means it is not caused by single microorganism. We cannot say that this particular organism is the only causative organism. So always endodontic infections are polymicrobial in nature. And when you see that, so we have in the beginning we have dealt about various types of organisms. We have said what is aerobic and what is anaerobic, right? So when you see in that terms, the endodontic infections are polymicrobial, and most of the infections are caused by the obligatory anaerobes. So root canal flora is predominantly by the anaerobic bacteria, and the facultative anaerobes are significantly in the coronal part, and the obligatory anaerobes significant in the apical part. Now in that anaerobic bacteria, 75% of the anaerobic bacteria is comprised of the gram-positive bacteria, examples being Staphylococci, Streptococci, Corynebacterium, and 25% would be gram-negative bacteria. Gram-negative bacteria, examples are Bacteroides, Neisseria, Fusobacterium. All these would be coming under the gram-negative, right? So again we are seeing the root canal flora usually is predominated by the anaerobic bacteria. In that anaerobic bacteria, we have 75% of them gram-positive, 25% of them are gram-negative. And in that anaerobic bacteria, facultative anaerobes are mostly seen in the coronal third, and obligatory anaerobes are usually seen in the apical third. So pulpal infections, as I already said, they are polymicrobial in nature, and when culture was done in an infected tooth, they said that the colony forming units which are seen in one single root canal ranges from 10 to the power of 2 to 10 to the power of 8, right? So many amount of bacteria can be cultured, can be extracted within a single root canal. Now you can imagine what is the amount of the microorganisms present in the endodontic infections, right? So majority of the microorganisms associated with endodontic infections are anaerobic in nature, and the anaerobic bacteria that constitute endodontic infections are obligatory anaerobes, constitute around 60 to 63%, as already told. Endodontic infections are predominantly obligatory anaerobes, and next is facultative anaerobes, which makes up to 34 to 36% of the anaerobic bacteria. Now the reason being why the facultative anaerobes are most commonly seen in the coronal fragment and why the obligatory anaerobes are seen in the apical fragment is, in the coronal segment high oxygen tension is present because it is there mostly on in the oral cavity, so the oxygen tension is more, nutrients are directly available from the oral cavity, and so because of the oxygen gradient and because of the nutrition present, so that is why facultative anaerobes are mostly seen in the coronal segment, right? And these bacteria which are present in the coronal segment are amenable for the endodontic procedure, that can be easily removed during the endodontic procedures. Coming to the middle segment, which is the main canal, so which is present in the middle third of the tooth. Here we can see gradually the oxygen tension decreases, and the amount of nutrients again are coming from the oral cavity itself, and the bacteria again are here can be removed because of the endodontic treatment. Now coming to the main aspect, which is the apical third fragment, right? So the apical third fragment, if we see, it is having very low oxygen tension because we are moving from the oral cavity and coming down into the alveolar bone. So the oxygen tension is very low, and the nutrients are usually coming from the periapical area, as contrast to the remaining two areas where the nutrition was coming from the oral cavity. Here the nutrition is coming from the periapical area, and here the most difficult part is that the microorganisms are not susceptible to the endodontic treatment. So that is why removing the bacteria from the apical third becomes very difficult, and because of the inability to remove the bacteria from the apical third, that causes most of the endodontic failures. So moving on to the endodontic infections, there are various types of endodontic infections. So the endodontic infections have been classified into intraradicular infections and extraradicular infections. Intraradicular infections are further classified into primary infections, secondary infections, and persistent infections. And so moving on to the intraradicular infections, we can see here they are divided into primary, secondary, and persistent. Primary again are divided into gram-negative infections and gram-positive infections. So these are the bacteria which are responsible for the gram-negative and gram-positive infections. Now we have intraradicular infection secondary, right, and these are the examples. So gram-negative falls under the examples of the bacteria which causes gram-negative primary endodontic infections are Tannerella, Dialister, Porphyromonas, Prevotella, Fusobacterium, Campylobacter, Treponema. Gram-positive includes Streptococcus, Eubacterium, and Actinomyces. And secondary infections are usually caused by Pseudomonas, E. coli, Candida, and E. faecalis. Persistent infections are usually caused by fungi and E. faecalis. So what is primary infection, secondary infection, persistent infection? Primary infections are those infections which are the main causative factors for the infection to cause, right? So the primary infections are the one which the bacteria which will cause the entire endodontic infection to happen. That is the reason why there is necrosis of the tooth. That is the reason why there is apical inflammation, there is apical periodontitis, are all mostly caused by the primary endodontic infection. So that primary endodontic infection we can be removed with the help of your endodontic treatment. That will be done. So primary endodontic infections can be treated with the help of endodontic treatment. Now coming to the secondary infection, okay, so we have seen that secondary infection is the one where it was not present in the beginning, right, but then gets later introduced into the tooth structure. For example, now a patient came to us, okay, with a completely asymptomatic tooth, okay, so there was no bacterial presence at all. But what happens is, because of the aseptic techniques, right, when we don't follow proper technique or during the treatment, we try to introduce the bacteria, so that causes secondary infection of the root canal system, right? So the secondary infection can be caused during the treatment, between the appointments, or after the root canal treatment. So the secondary infection is basically, it is not present in the beginning, but then it gets introduced during the treatment or after the treatment. So that is the secondary infection. Ideally, secondary infections and the persistent infections are usually difficult to get differentiated because they happen both together. Next is, so persistent infections are those infections which can be either primary or which can be even secondary also. These are the microorganisms, they usually resist any sort of intracanal medicaments. So because they resist, so they pertain in the root canal system. So the microorganisms that resist the intracanal antimicrobial procedures are survived in the treated canal. Involved microorganisms are remnants of either a primary infection or a secondary infection. Now here we can see this is the tooth, right? So after the endodontic treatment, so before the endodontic treatment, during the treatment, the bacteria is present. And if the bacteria is present after the treatment, if the bacteria is absent after the treatment, that means the bacteria have succumbed to the endodontic treatment. If the bacteria are present in the initial stages and also after the treatment also, this is called as a persistent infection. If the bacteria was absent in the beginning of the treatment and then gets present after the treatment, that is called as a secondary infection, right? So these are the various types of infections. So the primary endodontic infection, secondary endodontic infection, persistent endodontic infections are under the intraradicular infections. Intraradicular infections, that means the bacteria which are present within the root canal system. Now coming to the so how do you differentiate whether patient is having a persistent infection or a secondary infection? Usually it is very difficult to get isolated between the persistent and the secondary, but we have various symptomatic factors where we can say that the infection is persistent. That is, first is persistent exudation, persistent symptoms, intra-appointment flare-ups, failure of the endodontic treatment, and characterization of the post-treatment apical periodontic lesion. So these are all the factors which will tell us that the endodontic infection is persistent. So that you can see that the tooth is continuously draining. Sometimes the patient is complaining of continuous pain, and in the pain will be usually flared up from one appointment to the other appointment. So these are all the signs and symptoms of the persistent infection. Now the main important bacteria which is responsible for either the secondary infection or the persistent infection is E. faecalis. E. faecalis is one of the most common in bacteria which is isolated from the canals which have been cultured from the secondary or the persistent infection. Now what is the reason behind the E. faecalis to cause most type of infection is because it is having certain properties which other bacteria does not have, right? So the properties which are very much defined to E. faecalis are: it endures prolonged periods of nutritional deprivation. That means it can survive the periods of nutrition absence. That means it can also grow in the absence of nutrition. It converts itself into a spore form wherein it can be still alive in the case of nutritional deprivation. Next is one of the significant feature of the E. faecalis is that it can bind to the dentine properly. So it can bind to dentine, it can go into the deep areas of the dental tubules and it can get binded onto dentine so that it will not be subjected to the intracanal medicament. So that is the second most important feature of E. faecalis. Next is it alters the host response. That means it is going to alter the host immune response by not getting affected on its own. Next is it suppresses the action of the lymphocytes, that is the immune reaction of the patient. It possesses various enzymes. The bacterial byproducts are very, very harmful. Thus they decreasing the immunity of the patient, and it resists the intracanal medicaments, especially calcium hydroxide. Why? Because it has the ability to maintain the pH homeostasis, and also it competes with others. So these are the various significant factors which are very much specific to E. faecalis, which makes it very notorious for the secondary and the persistent infections. Now the Walfrid Fritsch in the year 1939 has categorized some zones of infection, zone A, B, C and D, depending on the microbial presence or absence. So he has named these zones as zone of infection, zone of contamination, zone of irritation and zone of stimulation. A is zone of contamination, zone of infection, where you can see n number of bacteria, which is a zone of infection is rich in all sort of infective bacteria. Whereas B zone is zone of contamination, where we can see some bacterial byproducts, right? We can also see the toxic reactions happening because of the bacterial byproducts. And zone A is completely filled with your polymorphonuclear neutrophils and leukocytes, where a zone of contamination is, we can see various inflammatory cells but not the bacteria. Zone C, so as we are moving out from the periapical area, the zone of infection reduces, the number of bacteria reduces. So zone C is a zone of irritation where we can see histiocytes and osteoclasts, where the bacterial toxicity reaction also will be less. And the last zone is zone of stimulation, which is zone D. It is basically the reparative zone, wherein because of the reactionary products which are produced by the bacteria, the body, the immunity will try to again start repairing, is that the zone D. So these are the areas called as zones of fish. One of the most commonly asked 4-mark question for your final year exam. Now coming to the other topic, which is called as biofilm. So what is a biofilm? Biofilm is a community of the microcolonies of the bacteria, which are usually characterized by cells that are attached to a substratum and are in a polymeric matrix substance called as EPS and exhibit altered growth phenotypes. So this is what has been changed the entire picture of endodontic microbiology. So biofilms have been isolated from the endodontic root in the root canals, and because of the property of biofilm formation, the bacterial infections of the endodontic infections become more resistant for the endodontic treatment. So how does a biofilm form? So biofilm forms in more 4 stages. So first is conditioning of the substrate, next is the addition of the bacteria, third is colonization, fourth is wherein detachment, the bacteria gets detached. So let us see the process of how the biofilm is formed, and we can see here first is the all the proteins get absorbed onto a substrate. So this is called as conditioning of the substrate. So once the conditioning layer is formed, then the bacteria will slowly come and attach to these protein substrates, right? So once the bacteria is slowly attaching, they try to form microcolonies, right, and also have coadhesion. Now once that these colonies are formed, after days and weeks, the bacteria will try to detach themselves, right? In the next picture we'll have a better view. So this is how it takes place. So first is this is called as conditioning of the substrate, right? So once the conditioning is done, the bacteria will come and settle down, the bacteria will slowly colonize and then they start and forming microcolonies. Once it is established, then all these bacteria will again try to detach. Some of the bacteria will get detached and again start the same process of biofilm formation. So this is how the biofilm is formed. We can see the attachment, colonization and growth, and the most important factor to happen this is because of the conditioning surface. Right. So what is the significance of the biofilm? Why are we supposed to know about the biofilm? So biofilm is important. Why? Because it has got these 4 important properties which will make it very difficult during the endodontic treatment. First is autopoiesis. Autopoiesis is nothing but wherein the bacteria can self-organize themselves, right? So this is one of the feature wherein they try to organize themselves so that they can accommodate space for the rest of the bacteria also. So this is one of the most important factor. Next is homeostasis. So they have the ability to overcome the environmental changes or challenges during the, so for example now we are trying to give some endodontic irrigating solutions in the root canal. So now this, because of the homeostasis, whatever the changes are taking place within the root canal system because of the endodontic irrigants, they will try to manage the environment and they will try to go against it. So that is called as homeostasis. Next is synergy. Synergy is that when an individual, they are weaker, but again when they try to communicate with the other co-fellows, then they try to have synergy. That means they are more powerful when they exist with their other bacteria. So that is the most important thing, that is synergy. And next is communality. Communality is when a problem comes, they face the problem as a combined unit and not as an individual. So these are the 4 most important factors which a biofilm possesses, and because of all these 4 problems, so because of the 4 factors, the removal of the endodontic infection becomes a challenge. So now these bacterial biofilms are also categorized into 4 different types depending on the location. So we have the 4 different types of the biofilms are intracanal biofilms, extraradicular biofilms, periapical biofilms, and biomaterial centered biofilms, right? So intracanal biofilms are the biofilms which are present exclusively within the canal, right? Extraradicular biofilms is they can be because of the extension of the intracanal biofilm. That means any intracanal biofilm which is exiting from the apical foramina and again starting a canal around the extraradicular biofilms, or an extraradicular biofilms can also can be developed on its own without the intracanal biofilm. So intracanal biofilm, extraradicular biofilm, periapical biofilm, where we can see the biofilm only at the apical area, not around the entire root surface. So that is the periapical biofilm. Next is biocentered biofilm, biomaterial centered biofilm. That is wherein sometimes when we see that we have various materials coming out from the apical foramina, that can be intracanal medicament like calcium hydroxide. It can also be gutta-percha which is coming out from the apical foramina. So when you have a foreign object, what happens? Again the same thing happens. There will be conditioning of the substrate. That is, for example, if we see gutta-percha, around the gutta-percha there can be conditioning of the gutta-percha surface and can cause colonization of the bacteria. So these are the 4 important types of bacterial biofilms what we see in the root canal system. Thank you.